Hi everybody, today we're going to learn about meiosis and sexual reproduction. So the first question that I want you to think about is why sex? Why do organisms do sexual reproduction? So there's a bunch of factors that contribute to variation among offspring, and that variation leads to populations being able to survive better. So crossing over you're going to learn about, and that results in recombinant chromatids, which gives you shuffled information from mom and dad. Random assortment, which we'll talk about, of how the chromosomes can line up, and also fertilization. So just to review with chromosome number, so remember germ cells are the cells that are at the beginning of meiosis, and they're diploid, so they have 2 of each chromosome. Gametes, which are the product of meiosis, your egg and sperm, have just 1 N, or 23 chromosomes in humans. So if you think about it, if a diploid cell has 24 chromosomes, that means it has 12 pairs. The gametes would each have 12 chromosomes. So here's a karyotype. This would be a female because there's 2 X's down here. And this would be before it doubles in synthesis. So meiosis 1 is where the homologous pairs separate. So it's a little bit different than mitosis, but in meiosis 1, the big thing is the homologous pairs separate. So these are homologous to each other. These would be sister chromatids. In meiosis 2, the big thing to remember here is DNA isn't doubled between the meiosis 1 and meiosis 2, and the 2 sister chromatids are pulled apart from each other. So in the beginning, before a cell goes into meiosis, it doubles its DNA. So it goes from 46 to 92. And then after meiosis 1, it has 46. And then after meiosis 2, each of those cells has 23 chromosomes. And there's 4 cells that are going to be produced. So we're going to go through the different stages of meiosis 1 and 2. And what I want you to do is I want you to draw the pictures that are below here, and I want you to label them with their appropriate phase. And then I want you to leave some room because we're going to talk about each of the phases. So go ahead and pause the video, draw it and label it, and leave some room for a description. Okay, so in prophase 1, certain things happen. The DNA condenses, so we actually see chromosomes here. The homologous chromosomes, so see how these are homologous chromosomes, they swap segments, and that's called crossing over, which we'll talk about in a little bit. The other thing is that the chromosomes start to attach to the spindle fibers. You can see the nuclear envelope breaking up, and then the centrioles, which are right here, are going to move to the poles, the 2 poles of the cell. So here's what crossing over is. So this is 1 and 2 homologous chromosomes, right, pairs. And what happens is they get zipped together, basically. And so they're non-sister chromosomes, so they're different. This is the one from, let's say, dad, and this one is from mom. And so they are going to get zipped together, and they're actually going to switch places. So you can see the product down here. And so now this chromosome right here is a completely new variation from what it was before. And this creates new allele combinations, which creates more variation, which is what we want in sexual reproduction. Okay, and this only happens in prophase 1. In metaphase 1, the homologous chromosomes go to the middle and they line up next to each other. That's really important because in metaphase 2, they line up on top of each other. And this is where what's called random alignment or random assortment happens. And what that is random assortment is the possible chromosome combination. So this is a figure to show you that. So your first combination could be all of these blue and light blue, okay? Or these 2 could flip, and now you have different combinations. Or 2 of them could flip, and then you have different combinations. Or the one in the middle could flip. So the way that you figure out how many different combinations it is For humans, it's 23 to the second. So there's 23 pairs, and there's two different ways those chromosomes could be. They could either be like this, or they could flip. Okay, so if you had um, 12 pairs, it would be 12 squared. Okay, and that's another way that we get variation. So crossing over in prophase 1 and random assortment in metaphase 1. In anaphase 1, homologous pairs are separated from each other, but the sister chromatids remain together. So that's important. Telophase, the chromosomes get to the poles, and then you get cytoplasmic division, which is either contractile ring or cell plate, depending on if you're an animal or plant. And then the nuclei form up here, and the nucleus forms down there. And then we move into prophase 2. So what happens here is the spindle fibers start to grab onto the chromosomes. Notice there's no crossing over here. There's no chromosomes condensing because they're already condensed. In metaphase 2, the duplicated chromosomes line up, but they don't line up next to each other. They line up on top of each other, basically, side by side. And they're lined up singularly. That's what I mean. They're not like in metaphase 1 where they're next to each other. In anaphase 2, the sister chromatids get pulled apart and they become independent chromosomes. And then in telophase 2, you get your 4 haploid cells. So here is just a picture of what the chromosomes actually look like in these different parts. So here is prophase 1, where you can see the chromosomes forming, metaphase 1, where they're starting to line up, anaphase 1, where they get pulled apart, and telophase 1, where they start to form another nuclei. And then prophase 2, right, they're starting to be grabbed by the spindle fibers, and they line up. Metaphase 2, get pulled apart, and then you get your 4 new gametes, each with half the information. So let's talk about egg and sperm formation. So notice that, so this is your primary oocyte is the germ cell, and the spermatogonium is the germ cell of sperm. So you don't need to know all these words. This is just regular old mitosis, right? One division here, and then another division, and the DNA is only replicated at the top. So the important things to know about this is with egg formation, only one egg actually gets produced and becomes mature. Then these 3 are called polar bodies, and they just dissolve. And the reason for this is because all of the organelles and everything needs to be in this mature egg because it's what's going to become the baby. So the purpose of these polar bodies is that they takes the extra genetic information because, right, you have 23, 23, 23, and 23 chromosomes for a human. And so that extra DNA here just is going to be broken down and only 23 will remain. So if you had more than 23 in the egg cell, then you would have issues with the development of a baby. Okay, if you look at sperm formation, sperm, you get 4 sperm, right? You don't just get 1 sperm, you get 4 sperm. And that's because the sperm deliver DNA. So they don't need to have organelles, they don't need to have a nucleus or anything like that. They just deliver the DNA and a tail so they can get where they're going. And so so all 4 of them survive. So a little bit about asexual reproduction. So this would be like mitosis, and there's a couple different ones you should know about. So parthenogenesis is where the embryos occur without fertilization. So females lay eggs that develop. Individuals are haploid. Okay, so the eggs are still haploid, and an example of this is whiptail lizards. So all of their cells are haploid, and their sex cells are basically just a regular cell that becomes a new lizard. Binary fission, we talked about a little bit before, and that's in prokaryotes where the bacteria where the DNA is replicated and the cell just splits. And then budding is where a new organism grows on another one until it's developed. And it's identical to the clone. So some plants do this, like aspens do this, fungi, and then hydras, which are a part of a jellyfish lifestyle. And so something that can go wrong in meiosis, you might have remembered it from biology, is called non-disjunction. So when the chromosomes are being pulled apart, if they don't get pulled apart correctly, you could get an extra one on one side and not enough on the other side. And that can lead to a monosomy, which is one, or it could lead to a trisomy, which is three of something. So if one sex cell is missing a chromosome, then when that sex cell gets together with another one, it's going to have only one of those, let's say, number 2 chromosomes, right? Because you always get one from mom and one from dad from the egg and sperm. Now, in the cell that had 2, so the sex cell, the egg cell that has 2 number 2s, would maybe get together with another cell from the sperm that has 1, and so then you would have 3 number 2s. So, um, for example, this one has an extra 21, and so that would mean this would be Down syndrome, and so that would be a trisomy. Okay, and the last thing that makes more genetic variation in sexual reproduction is fertilization. So the male and female gametes come together and make one nucleus, and it's totally random which gametes are going to come together, and that adds to variation. And when those two haploid nuclei come together, so you get N plus N and you get a 2N, that's what a zygote is called. And again, it's totally random which egg and sperm come together, and so that's the third thing that leads to variation. So you have crossing over in prophase 1, where the homologous chromosomes cross and exchange material. Then you have random assortment in metaphase 1, where the chromosomes could line up like this, or they could line up like this, and then you have 23 different combinations, so it's 23 squared. And then the last thing is fertilization, where it's totally random which gamete and which egg and which sperm come together. And that's it with meiosis. Hope you have a great day.